Assalamualaikum, hi and good day to all the lecturers and examiners. My name is Dayang Amira binti Abang Halim Badar Sham 72179 and my supervisor for my final year project is Dr. Ngeng Singh. Today, I'm going to present my FYP entitled Antimicrobial Activity of Endophyte Marasmius cladophyllus UMAS MS8. My presentation comprises of 7 elements which are introduction, objectives and hypothesis, literature review, materials and method, expected outcome, work schedule, and references. First and foremost, this study will be done in order to find the alternative chemotherapeutic drugs in disease treatment due to the increased resistance of microorganisms caused by increased usage of antibiotics among people and various side effects caused by the medications. It is also my problem statement for my study. Mushroom has been chosen to be an alternative medication due to the traditional usage of mushroom as the medication. In this study, I will use Marasmius cladophyllus as the mushroom source. Mushroom is a plant source that is high in nutrients and help promoting vitamins, minerals, and antioxidants. There are 2 main objectives in my study. Firstly, it is mainly to screen the antimicrobial activity of Marasmius cladophyllus. Besides that, it is also aims to determine the minimum inhibitory concentration or MIC and minimum bactericidal concentration MBC of the fungal extract. My hypothesis for this study is Marasmius cladophyllus may be an effective antimicrobial agent in killing bacteria and yeast. For my literature review, I will talk about overview of mushroom, taxonomy of Marasmius cladophyllus, mushroom usage in medications, mushroom is antimicrobial agent, and antimicrobial activity of Marasmius species. For the first one, which is overview of mushroom. Mushroom has long been known in all over the world as a medicine or therapeutic food among the rural people. According to Waktola and Temisgen 2018, this plant source can prevent diseases like hypertension, hypercholesterolemia, and cancer due to the content of the mushroom itself such as high fiber contents, proteins, microelements, and low calorie content. As we can see from the picture attached, it shows a hypertension and a laboratory test results that shows a high value of cholesterol. Mushroom also has 8 amino acids, polyunsaturated fatty acids, and a small amount of saturated fatty acid. As we can see from the table, these are the examples of 7 medicinal mushroom that has been used in medications. Next, I will talk about taxonomy of Marasmius cladophyllus. According to Kirk 2020, Marasmius cladophyllus is a species of fungus that belongs to the order of Agaricales. It is from the phylum of Basidiomycota and belongs to class Agaricomycetes. To be specific, Marasmius cladophyllus is belonging to family of Marasmiaceae and genus Marasmius. I will talk about mushroom usage in medications. There are numerous of therapeutic applications of mushroom which are antimicrobial activity, anti-cancer activity, anti-asthmatic activity, hepatoprotective effects, and enhancing immunogenicity. Mushroom has this therapeutic potential due to the existence of few types of secondary metabolites such as alpha-beta unsaturated polysaccharides, glycoproteins, peptides, phenolic derivatives, hydrolytics, lipids, and oxidative enzymes. Polysaccharides are the most important secondary metabolites that have a cancer properties and it can enhance the immune system. Furthermore, mushroom also can be used as antimicrobial agent. As we can see from the figure, confluentine, grifolin, and neogrifolin are the compounds that have been identified in the Albatrellus flaty. It showed an antimicrobial activity against Bacillus cereus and Enterococcus facilis. On the other hand, Genomycin A and Genomycin B also have been identified from Ganoderma piferi. These compounds showed an antimicrobial activity against Bacillus subtilis, Micrococcus flavus, and Staphylococcus aureus. From there, we know that antimicrobial activity is referring to all the active principles or agents that used to inhibit the bacteria from growing and prevent the formation of microbial colonies. Hot water extraction also much preferable than alcohol due to the polarity of the components. For antimicrobial activity of Marasmius species, there is a research demonstrates that the ethanol extracts of Marasmius oredes showed an antimicrobial activity against gram-positive bacteria such as Enterococcus faecium and Staphylococcus epidermidis. However, it does not show any antimicrobial activity against gram-negative bacteria. Besides that, there is an antimicrobial activity of Marasmius oridis methanol extract against various gram-positive and gram-negative bacteria, including Pseudomonas aeruginosa, Staphylococcus aureus, Staphylococcus epidermidis, and Escherichia coli. Furthermore, methanol extracts of some wild mushroom were discovered to have a higher antimicrobial activity and give the effect on nosocomial pathogens than typically used antibiotics. These findings show that other mushroom products can be used instead of antibiotics in the treatment of infection disease. Antimicrobial activity screening of Marasmius cladophyllus therefore might enable a better understanding of the antimicrobial potential of the cladophyllus species that have not been studied. I will proceed my presentation with materials and method. For materials, I will be using 96-well plates, ethyl acetate, filter paper, gentamicin disc, glycerol, inoculation loop, mule-hinton agar, mule-hinton broth, sterile cotton swab, trypticase soya broth, mushroom which is Marasmius cladophyllus, and sterile distilled water. For the methods, it consists of 5 main methods which are microorganisms preparation and culture maintenance, bioactive compound extraction, antibacterial assay, and determination of MIC and MBC. For the first method, which is microorganisms, Marasmius cladophyllus will be obtained from the Molecular Genetic Laboratory of Faculty of Resource Science and Technology UMAS. Candida albicans, Escherichia coli which is gram-negative bacteria, and Staphylococcus aureus which is gram-positive bacteria will be used to determine the antimicrobial activity of Marasmius cladophyllus. For the second method, which is microorganisms preparation and culture maintenance, Marasmius cladophyllus will be grown in mule-hinton agar plates for 7 to 10 days at 34 degrees Celsius in order to have a fungal source. Stock fungus culture will be maintained in 20% volume per volume glycerol and will be stored at negative 20 degrees Celsius in sterile distilled water. Several fungal agar plugs from the MHA plates will be taken and grown in trypticase soya broth for 10 days at room temperature with shaking 150 rotations per minute. Next, I will proceed with bioactive compound extraction. The liquid broth will be filtered in order to separate the fungal mycelium from the media. Upon filter, 2 samples will be produced, which are fungal excretion compound sample A and fungal biomass sample B. Both samples will be extracted with ethyl acetate. However, sample A will be extracted in a separating funnel, while sample B will be extracted in Socklet apparatus. Once done, the ethyl acetate layer will be separated and dried by using rotary evaporator. For antibacterial assay, this division assay method will be used. Bacterial assay inoculum will be adjusted to 0.5 mcfarland standard turbidity. Bacterial inoculum will be used to make a lot of bacterial on the MHA plates. Both samples will be tested for antimicrobial activity. Filter paper disc will be soaked in both samples and gentamicin. Gentamicin will be utilized as a control. This filter paper will be placed on the top of the nutrient agar. This method can be referred to the figure attached. The antibacterial assay results will be determined by observing the halo formation after 24 hours and measuring the zone of inhibition diameter on the agar as shown on the figures day. For the next method, which is determination of MIC and MBC, for minimum inhibitory concentration, which is MIC, broth microdilution method will be used. The fungal compound will be prepared 100 ml. Inoculum will be adjusted to 0.5 mcfarland standard turbidity as well, then will be diluted to 1/150 times. 100 microliter of the compound will be transferred to the first well. Two-fold dilution will be done by transferring half of the compound concentration, which is 50 microliter, in the first well until the 11th well. Gentamicin will be placed in the 11th well and it will be utilized as a positive control. Broth alone will be placed in the 12th well and it will be utilized as a negative control. MIC will be determined by observing the lowest concentration of the fungal excretion compound that shows no precipitate present in the well. For minimum bactericidal concentration, which is MBC, 100 microliter of the broth will be added on the top of the nutrient agar plates. 100 microliter gentamicin will be added and it will be utilized as a positive control. MBC will be determined by observing the lowest concentration of the fungal excretion compound that shows no bacterial growth on the nutrient agar. We have come to the next part of my presentation, which is expected outcome. Fungal excretion compound and fungal biomass of Marasmius cladophyllus may show an antimicrobial activity against the tested bacteria and yeast, which are Escherichia coli, Staphylococcus aureus, and Candida albicans. The fungal extract containing the extracellular compounds may give a higher antimicrobial activity against the bacteria and yeast. Minimum inhibition concentration, MIC, of the fungal excretion compound may be lower than minimum bactericidal concentration, MBC, of the compound. It is because MIC is only needed to inhibit the visible growth of the bacteria and yeast, but not all of the bacteria in the concentration are dead. Thus, MBC will be done to know the exact concentration that will be used to kill 100% bacteria. This is my work schedule. I started to choose my FYP title and confirm it in October 2021. I proceed my progress with data collection in October and November. Then I started to do my proposal writing in November and December. I presented my FYP proposal and submitted my proposal on January 2022. I will start my FYP 2 and do all the lab works in the middle of January until May. Report writing and report presentation will be held in April to June. These are the references that I used to look for the information in my presentation. That's all for my presentation. Thank you.